Okay, thank you all for coming today. I really appreciate it. We're so excited about having a discussion about egg assessment. Having spent most of our time discussing embryos, we realize how important the egg is. So if you're going to talk about eggs, you really cannot do that without Laura Rienzi. Laura Rienzi is really the source of all the knowledge that... It was really the beginning of having discussions about egg quality. The major publications we have around egg assessment and the attempts, which really stemmed from the challenges that you were experiencing in Italy as well. So it was a great honor to have you here today.
My honor, my pleasure.
We would also like to invite Roberta Maggioli onto the stage. Roberta is the Director of Operations in Genera Life, and I'm really looking forward to having this discussion with the two of you alongside Asaf. And yeah, let's talk eggs.
Okay, so first of all, we just want to know a little bit what is your personal interest in eggs. I know that you did a lot of research about eggs. Okay. Yeah.
So it's quite funny because we were obliged to look at eggs when a very severe law came in force.
Sorry, I think we have
Sorry for that. Can you hear me?
Okay, can you guys hear sound back? Nikika? Nikika Zaninovic. Nikika Zaninovic, hello! Yes! Thank you guys. Okay, over to you.
Well, I was just saying a story because
Yeah.
sometimes bad things turn to be something really, really incredibly challenging. So there was a very bad law in Italy where we were obliged to inseminate only 3 eggs. So imagine, instead of being able to make fertilization on all the eggs that we were collecting from patients, we had to choose 3 eggs. And it was in 2004, and there were no information about quality of eggs. We were all postponing our assessment at the embryo of a blastocyst. So we start looking to the eggs. We start looking how to choose the eggs, and it was so difficult, but up to date. We don't know how to choose eggs except for very few information. So this is why we are looking so much about understanding the quality of eggs, because time has changed, not anymore because of the law. The law finally was removed in Italy, but because we have a new population of patients that needs to know the quality of the eggs that we are preserving for them in fertility preservation, or we are assigning to them during egg donation program. So back to the future, we have to really know to predict the quality of the eggs to be able to enhance our programs of fertility preservation and egg donation.
So, what kind of parameters did we use till today, the embryologists use till today to assess the eggs quality?
So we mainly focus, we still mainly focus on oocyte morphology, so the morphological appearance of the oocyte, because we know that there are some sophisticated and probably more comprehensive technique to evaluate the quality of the oocyte, but most of the limited the issue that we have in applying this technique then in the clinical practice is that those techniques are usually time-consuming, costly, so require also highly training embryologists because we can require the extra manipulation if we think, for instance, to the repetition genetic testing in polar bodies or even the use of polarized light microscopy. So require extra instrument manipulation. So basically are technology that are costly and time consuming. Usually they cannot predict the quality, they cannot give us insight about the quality of the oocyte. So I think the main limitation is related to the cost effectiveness of this technique. Probably in the future we will have a combination of this technique, because there are some techniques very promising, such as, for instance, the biomechanics, the metabolomics are all non-invasive technique. It can be very promising, but at the moment it still need validation. So at the moment we are still focusing on just looking at the oocyte, but we are not selecting at all, because in order not to impair, to reduce the efficacy of the treatment, we used to inseminate all the oocyte that we have available, because we are not able to predict the quality of the oocyte.
What?
If I may say, in the lab, something will work if it's easy, friendly for embryologists, and cost effective.
Okay.
So all the technology that we have over the morphology needs instrumentation, know-how, is not understandable, is not really validated. So we need something that in the lab is really changing our life, and it has to be fast and easy.
Can you tell us a little bit about the ESHRE guidelines about oocyte assessment?
Yeah, so I'm very proud to be part of a group that is revising the ESHRE Alpha guidelines for oocyte, DM, embryo, and blastocyst morphology. You know, the Istanbul consensus. It's very important because we need reference value to understand the quality of our labs, to look to our KPIs. Roberta is looking to KPI every week, and it's very important to have references. And we have made an incredible job this time, because we have a different approach, which is much more systematic, with meta-analysis. Although meta-analysis are not the best, we expect much more for artificial intelligence also on
we'll
uniform data.
talk about it, yes.
That's for sure, but to date, we still have, for the last time, I hope, we are working with meta-analysis, so we are going to try to advise which are the reference level that we have to reach in fertilization rate, in how to assess VMO, how to assess the blastocyst, and how to assess VX. And today there are very few that we could really state that is useful for egg assessment. So there is a lot of room for improvement, guys.
Yeah, I agree. So when it comes to these challenges that we're experiencing with egg assessment in particular, what are the strengths within the AI technology that you think that can help solve some of these challenges?
I think that the world is changing, and as it was said before, we have to look to the future, and to rely on that, on data, will be so important for every single step of the IVF procedure. It has been said by NHS that medicine was a science supported by data. Now it will be a medical science work in collaboration with data scientists. It's not anymore that we look to the data and we try to find a correlation or causality. More often we do a confusion between the two, but we have data scientists that will guide us in the decision making. There is no doubt that there is no doctor that has the same experience as a big database. The best doctor has seen 10,000 patients. A big database has 10 million patients inside. So I'm sure that for every step of the medicine in general, of our life in general, and also in the lab, we are going to rely on big data. And of course, we are also going to give data with machine learning, will further increase our capacity to understanding what we are doing. The quality of data will be very important.
What do you think, Roberta, in terms of
Yeah,
how
everyday
AI
work.
is coming in? What are your thoughts on
I'm
AI?
working a lot on standardization, so I think that is one of the short-term strength that the introduction of AI may lead to the lab, since we still have a lot of subjectivity, a lot of interoperatory variability in the assessment of the morphological assessment of the oocyte. So I think the short-term strength is the improvement of the standardization and the reduction of the variability between operators. It's something that is challenging now for us to evaluate also the performance of the center, the KPIs, because to identify also to standardize criteria for oocyte assessment, because there is a high grade of subjectivity in the evaluation. So I think this is one of the main points.
Okay, so we're gonna use data.
Yeah, of course.
And,
We're looking for.
How the data should look like? So we should look on pre-ICSI or post-ICSI oocyte, or do you think there's no any difference, or there is of course advantage of pre-ICSI oocyte images?
It depends on the patient indication. I think we need everything because the need of the patient is changing, and as I said, for fertility preservation, we need to know on pre-ICSI. For IVF, I think we need the whole journey of the embryo in our labs. But of course, the outcome measure has to be the live birth. Because if we stop our algorithm to the blastocyst formation, which is actually the currency in IVF, you know, how many blastocysts do you have, how many blastocysts do you produce? But at the end of the story, we want a baby. Our patients want a baby, and our gynecologists are asking for a baby. So it is very important to validate whatever will be on the live birth.
Yeah, no, I agree. But the challenge that we have here is that if we go with live birth, how many confounders do we need to control for? Maybe you could comment with us, what are the confounders that we need to control if we're looking at blastulation or ploidy or live birth? When you're looking at the egg, which is, well, only part of the story, we've got to have the sperm and the uterus and, you know, the environment and so on. So how would you, what are the high-level confounders that you would want to consider with the different endpoints?
If you look to euploid blastocyst transfer, the confounder disappear a lot. You know? I mean, with the euploid blastocyst transfer, with very few exceptions, which is normal uterus or high BMI, we have a very stable implantation potential. No? So it's clear that the confounders are there. We don't have 100% live birth with but it is an excellent model that we can use also to assess different strategy. Before euploid blastocyst transfer, the confounder were really a big problem. With this model, we can work on it.
So I'm thinking now in the scenario of you have a good egg but a bad sperm, for instance. So the tool's okay, but we put the bad sperm inside, and therefore no blast was formed, no baby was formed. What are your thoughts on that?
According to our studies, the bad sperm is going to have a great influence on fertilization and blastulation, but once you have a blastocyst and euploid blastocyst, the male factor in fertility disappear.
Okay.
So we have to work on bigger data. This is our study on, I think, 2,000 cycles. We have to work on more cycles, but we have to find which are the confounders. And I think big data will help us to find the real confounders.
Okay.
I think we all agree that live birth is the final outcome, because this is the goal, this is the main goal of IVF. We are not looking for blast, we are not looking for a positive HCG test, we are looking for the baby. But on the other hand, the problem is that if we will look on live birth, it will take years to make those studies. I'm not talking about the money, I'm talking about time. And until the end of the study, it will not be Chloe OQ version 1.0, it will be Chloe 6.0.
I know.
So it will be irrelevant for the future. So
we can start with blastulation rate, which
Okay.
is, as I said, the currency in IVF. But I think that we have to extend it and validate it.
We have to aim beyond.
Yeah.
Yeah.
I mean, it's a good start.
Mhm.
You know, when we do egg donation, what are we talking about with the patient? We ensure 1 blastocyst, 2 blastocysts. So it has a value also for the everyday clinical application. But then we have to validate on live birth, because otherwise we will not be, the expectation that we give maybe too high.
So Roberta, I'd love to hear your thoughts on the challenges with regards to traceability. So once you assess an egg, and you know that the egg is scored, whatever, how do you ensure that this is the egg that becomes that embryo, that becomes that one transferred, and so on?
Yeah, I think this is one of the biggest issues that we may have also after introduction of AI algorithm, because I mean, even if you can acquire images at the moment of the ICSI procedure and then inseminate and keep the tracking of the single oocyte by using a single drop
Okay.
culture, because it's the only way that we can assure traceability. But I think, we still rely on the operators. So until we have an automatic system that can automate completely all the steps of the IVF process, and then we can really be sure about the traceability of the images acquired, follow all the steps of the development. We have to be sure that, because it can be an issue. So it can, I think, impair the accuracy of the software, the algorithm, because if the approach is not so accurate in keep tracking the picture, it can be challenging.
So you've been quite big on witnessing as well.
Yeah,
Every time we're big on many different topics.
Yeah, but it's the same problem that we have with the PGT cycles. Witness, also the automatic system that we have, it can help us in tracking the identity of the couples, but then we need a second embryologist to track the single embryo. So.
And when you cryopreserve eggs, how many do you put on a straw, for instance?
Up to 3?
Up to 3.
Up to 3.
She does. I don't remember anymore.
Was that a question?
Yeah.
What's a straw?
So up to 3.
Up to 3, but up to if it is a fertility preservation cases. Also, yeah.
And in those scenarios, is there a solution for you've assessed an egg and then now you put it into a straw, and then how do you maintain we don't have a solution for that at the moment, do we?
Yeah, at the moment we just, if you have, for instance, in the oocyte donation cycle, if you have a different donation on the same donor, we keep tracking of that donation, but it's difficult it's time consuming for the we can
I
do
have a
it.
solution. If the computer will recognize the oocyte, we can make a picture before vitrification, and then automatically it will be recognized for.
So if you have the library of pictures from before, you might be able to compare them after. Let's say there's a issue with the zona. Is that what you're thinking?
Whatever. Each oocyte is different from the other, so I'm sure that if we have a software that is looking for the oocyte, it will recognize the oocyte.
Right.
This is the future.
But to do that, you have to have a library of images first, right? So that you can
We
do
have to have a
this
software
comparison.
that is looking, but is tracking each oocyte, and then when we put back the oocyte with that code, he recognizes up to the. We are just talking brainstorming.
Well, it's not quite the future, as we're going to see very soon. It's the
Really?
present.
So it's not my idea. I had it already.
And beside the traceability, where do you prefer to do the eggs assessment? Inside an incubator or under the microscope?
Today, it's under a microscope. It's the only way we have. Of course, my dream would be that all my reproductive cells will always be in an incubator.
Okay.
That's for sure. Because honestly, the most important thing for the young embryologists is the culture system to ensure that you are not harming your cell. So if you have to choose to use a tool outside the incubator, leave the oocyte inside the incubator, remember that each time I will take them out, it has a cost. It potentially harms your cells. So culture and traceability are the basic of embryology. Ensure a good culture system and a perfect traceability. Then you can start to make ads on and things like that.
Can you talk a little bit about what are the harms that the egg can be exposed to, biologically speaking, when outside an incubator?
Yeah, so I put a picture in all my lab, which it's a figure that comes from Gardner and Whale review on Neuron Production Update. It's like the Bible in IVF. And the picture is like that. The oocyte is big like that, then you have the embryo, then you have a blastocyst. And this is the sensitivity of the cell to the in vitro culture, to physical, chemical things that happens in the lab. So the oocyte is big like that, very sensitive. The blastocyst is small like that, able to react to physical and chemical changes. And the oocyte is so sensitive, not because it's big, because 2PN is also big. It's because it has a meiotic spindle, which is sensitive to temperature, because it has a very big cytoskeleton that has to support PN formation and migration. So when you touch an oocyte, David Gardner used to say, it's like a baby naked outside. Then little by little you put clothes on it. So that's why the oocyte is the cell, and also epigenetic issue. We have a lot of different issues with oocyte that we don't have in the cleaving area in the blastocyst. So this is where we have to be pay more attention.
It's unfortunate, isn't it, that we have we don't have many ways of improving. We don't have any ways of improving the egg, but we have a ridiculous amount of ways to harm it.
Exactly. We always say that.
Best case
No
scenario,
therapy.
maintain the same.
Best case scenario, we fully exploit the quality of the eggs and the sperm that the patient is giving us.
And that's kind of our responsibility, isn't it?
Yeah, to
Roberta?
preserve. We have to preserve the viability.
Excellent. Any other thoughts you want to share?
No, I'm very excited to see what will be the future.
Okay, you made it.
We'd love
You
to
made it?
We'd love to open it to, if anybody has any questions to these two lovely ladies. You don't get much more of an expert than this. Any questions from the audience? Please, Liliana.
So, do you think that the oocyte assessment should be different for the oocytes coming from the patients and the oocytes coming from the donors?
So what I think is that the patient, the data on the patient is the most predictive, is where we can learn about the oocyte quality. So donors are potentially the best patient that we can have, but they are human. They are not machine, they are not factory. So probably we have to go deeper also, as we do for infertile population, we have to go deeper inside the anamnesis of the donor, because we know, Roberta is presenting here in ESHRE a poster showing that 7% of the donor are not behaving how we expect. And we know that there are some genes that are associated with that. So probably we will have to study the donor as we study the infertile population to be sure to target the number of oocytes to be assigned not only to the aspect of the oocyte, not only to the ovarian reserve and the age, but also to many other parameters of the donor itself. So I consider a donor woman exactly like a patient that is doing an IVF.
Okay, so the morphological assessment is same, but the inclusion criteria still should be different and more advanced for the donors.
Or we can target the number of eggs according to what we know about the prediction on blastocyst formation and live birth that we can have. Imagine that you have an excellent donor, we can assign 4 eggs. Imagine that we know that that donor has a slightly lower blastulation rate expected, then we assign 10 eggs. I mean, donors are very precious. I would not exclude a donor because maybe she has a slightly lower fertilization potential. No, I mean, this is part of our duty to fully exploit also what the donor are giving us.
Thank you.
Do you think that some patients have oocytes more cryosensible to freezing?
Very, very good question. I want to ask them. We have to find biomarkers. I mean, the oocyte was not built to be vitrified. Vitrification is incredibly efficient. It's fantastic. It has changed everything in IVF, but still, there are some quote of oocyte, although very good if inseminated fresh, but are more sensitive, of course, to cryopreservation. It's something that is related to the membrane permeability mainly. So why not to find something where we can assess the membrane permeability or some mechanical factors that will predict the survival rate. So
do you think that can be because some membranes are less cholesterol than other membranes?
can be for many different reasons. But membrane permeability is probably and also the sensitivity of shrinkage, so related to microtubules and microfilaments. So there are many different aspects that we can study. We are lucky, the majority of the oocyte, super good survivor. In other species, the survival rate is lower, but still we can have some quote of oocyte more sensitive, and not because they are of worse quality.
Okay, thank you.
Good question.
Any other questions from the audience?
Thank you very much. We have 2 questions. The first one, since Italy has a great experience in vitrifying eggs in your clinics, what would be the survival rates for patient eggs and for donor eggs? Is the first question.
Roberta, she knows all the numbers every week, so.
Our benchmarking for donor eggs is about 93-94%. For donor eggs is a little bit lower for for infertile patients eggs, and we expect to reach like 90-91% as benchmark KPI. We always monitor the survival rate on a monthly basis, and we have our graph where we monitor. I mean, we build the graph.
So in the last 2 years, we moved from 85 with all the adjustment that we did. So
small adjustments.
yeah, it was very funny because we are a big network, and there was one little center somewhere, I don't say where the survival rate was significantly higher than all the other centers. We were using exactly the same thing. So Roberta went there. She spent 1 week and she could not find anything except stupid things. The stupid things were implemented in the other center, and we moved from 85 to 91.
91, 92.
And 6% is a lot.
Was for patients' access, yes.
For donor eggs,
for eggs,
because
and
we
then
were
for-
using the same bank, so I mean the results were expected to be the same in.
So the difference was significant also in donor bank, and we use the same bank, and probably we use sometimes the same eggs, but we're split it to two centers. So we had the perfect model to understand that there was something there that was done. And it was related to volumes, to dimension of the pipette, or to timing, very, very small things. I mean, I am the one that has set the vitrification protocol. I say, I know my job. No, I didn't know. Because little by little you make little mistakes, you change little things in your lab, you don't realize. And to have experience, so go around, see centers, we are all open. I mean, I have visitor every week, we have visitor every week. Because looking to what each time I visit a center, even the worst center in the world, I find something better than what happens in my lab. And probably sooner or later we will have a guideline saying, and try to do something similar in all centers, that is really welcome.
So, yeah, 6% is a lot, but you said the stupid thing. What was the stupid thing that you changed?
The stupid things was the volume,
The standardization. It is a very small group, small team. They work exactly in the same way. So there is a high grade of standardization in that lab. So they are very precise, and I mean this is one of the main things that make the difference.
And the volume.
Then the volume. I mean, it's the volume of the sewing solution that they use. That is a higher volume, but it's not the concept that you need a higher volume to maximize the survival rate. It's just that by using a higher volume, you can minimize the
you can balance some little mistake, but I mean, it's a manual
Higher
procedure.
the volume, it's like slower the cooling, right?
The
The
sewing
following of the
solution.
TS medium, right?
The sewing solution. So
Going,
they use
going, TS.
the
So
double
the TS, no?
yes, right? They double the volume, the ml. So they use 4 ml for each procedure, and they just were able to achieve the highest
So the
results.
idea is that if you use higher volume and you do little mistakes in timings and things, or you prepare too early your dish, the evaporation will be less, of course, less impacting. If you have the high volume, you can really standardize for morning to night everything.
Okay, well, thank you very much. If you have any more questions, Laura is here, and you can have a chat with her directly after, and so is Roberta, who can help clarify some of these questions. Thank you both so much. It's been incredibly exciting.